Okay, so this is Psych 3280 for week 7, Neural-based subconsciousness. And this week, we are going to talk about higher visual areas and NCC. So just to start from the recap of the last week, week 6, we first introduced the concept of the NCC, neural correlates of consciousness. And that is the minimal set or a set of neurons that is sufficient to give rise to a particular conscious person. And this is a hypothetical entity in the brain, and here a particular conscious percept may be something like visual motion of particular direction, particular speed, and so on. And then we also introduced empirical NCC search strategies from the Chris Frith's paper in TICS. There, it articulated that it is most efficient to do that by fixing either sensory input or conscious percept or behavior output while varying the others. And then I went through all the details of the tables. And I suggested that utilizing this concept would be very useful for your final writing assessment to propose a new empirical research for consciousness. Okay. Also, I covered several topics as well in last week. One is that whether the NCC can be found in V1. And there are several reasons to say that V1 is unlikely candidate for the NCC. And one of the reasons is that non-conscious orientation dependent adaptation using this demo of the crowded orientation stimulus. I tried to show that it is likely to be mediated by V1, but this adaptation does not require conscious experience. Therefore, V1's activity that is sufficient to generate this adaptation is not really sufficient to generate conscious percept of this orientation. And the other arguments include that the dreaming suppresses V1, or blinks and eye movements are still is reflected by the V1 activity, but our conscious experience usually do not reflect the eye blink or eye movements. Okay. So that's the one of the several reasons for V1 as unlikely candidate for the NCC. All right. And then on the other hand, I argued that MT, middle temporal area, responsible for motion perception, is likely to be also the neural correlates of motion qualia. And the reason, one of the reasons is that motion after effects, that after adapting this, expanding motion stimulus, the Buddha stimulus, stationary Buddha stimulus probably felt like shrinking or the other way around. And these are mediated most likely by empty neurons. And for this type of motion after effects to work, it requires visibility of the motion stimulus, especially for those complicated motion type. And most likely, these motion after effects are mediated by more empty neurons. So both, after effects are requires MT and MT activation inferred from motion after effects are sufficient to generate motion qualia. Also, I reviewed some of the literature on the lesions of bilateral MT, which is very rare, abolishes motion percept and make the life almost like stroboscopic or disco-like experience. And also neural recordings done by the Newsome teams in Stanford and other US areas are very convincing that momentary fluctuation of the neuron activity correlates with monkey's perceptual report, especially with the random dot motion, and also microstimulation of the MTs by virtue of the MT being the columnar organization, also biases monkey's perceptual reports. And taken together, all these imply it's likely that empty neuron activity and so connectivity is sufficient for motion qualia. So this week, week 7, we will review the evidence for higher visual areas as the candidate for the other content-specific NCCs. And I will introduce powerful tools for consciousness research. Okay, and that includes binocular rivalry and continuous flash suppression and the decoding of the neural activity. So in terms of the learning objectives specifically, I'm going to, you are supposed to be able to answer the question of what are the evidence supporting the claim that higher visual areas are the NCC. And for this matter, the Quest for Consciousness chapter 7 and 8 are the recommended readings. And also, what are the phenomenological and behavior characteristics of binocular rivalry? And how did the researchers establish a binocular rivalry paradigm with monkeys as participants? And what are the implications of the NCC studies using binocular rivalry? And for that matter, Quest for Consciousness chapter 16 is going to be very useful. Okay, so let's first start with the V4 as the first target areas of the higher visual areas candidate for NCC. Okay, V4. So as I reviewed in week 5, each areas have a distinct connectivity pattern in the laminar structure. So if it is receiving the forward input from the lower areas, it tends to receive the input at the middle layer, layer 4. And then if it receives the feedback connection from other areas, it tends to receive the synaptic input at the lower or upper area of the layers. And based on this type of the layer structure and also connectivity patterns, people have constructed this Van Nessen diagram for the visual hierarchy. And so far, we have basically covered the retinal neurons, LGN, lateral geniculate neurons, and V1 that starts to possess the orientation selectivity, and then V2 for the illusory contours. And from here, as you can see, the diagram becomes very, very complicated. So V2, after that, V3 or V3 dorsal, V3 ventral. So this left side is more like a dorsal areas, the areas that goes the side of the back of the brain. And then this right side goes to the ventral. Ventral means, on the very side of the brain. And here, the MT locates more or less intermediate between the ventral and dorsal. So that's here. And that's what we focused last week. And then V4 locates into the ventral ventral areas and around the same hierarchy as MT. So V4 is still a part of the retinotopic area, means that the input from the outside visual areas, visual input is more or less faithfully represented in terms of relations. But remember that all of these are distorted in terms of the cortical magnification and the receptive field size. So fovea of the left visual field is represented in this all right hemisphere, okay? All of this is the right hemisphere. And for monkey and human, and it's compared. And then right hemisphere is unfolded. And if you don't understand it, you go to week 5 lecture video again. And then the left visual field upper part is represented by the ventral part of the V1, V2, and then V3, V4. And then this side ventral part, this is a ventral, and then this is a dorsal part. and then MT is intermediate, is represented many times. Okay, and then V4 is a part of the ventral areas, and this has been suggested as an particular neural correlates of consciousness before. So one of the most compelling evidence for the V4's involvement of the consciousness has been this color qualia. And here I'm going to review one of the case reports of the patient AR, who was studied around 2000 by Jack Gallant's group. And here, again, this is a radiological convention. So the right side of the brain is represented in the left, and the left side of the brain is presented in the right side. Okay, and this triangular points the highly restricted lesion in this particular patient. And this patient, after some lesion, they lost some color perception. Okay, and however, initially, and also even after he was tested with a clinical trial, he's quite remarkably, as Crystal Cox's book says, that this patient and others like him do not notice that they see in gray in one part of the visual field and in color in another. So here, this AR's case is the right and the ventral area, and that corresponds to the visual field of, this is the left visual field, so this is a fovea. Upper part of the visual field is affected by this one-sided brain lesion, and as a result, he was tested as achromatopsia, or loss of the color only in here but not other area. That's very interesting situation. And the more interesting thing is that this patient do not spontaneously notice that it is gray. And this is actually a highly profound kind of concept in consciousness research. If you don't have a capacity to experience something, you actually do not notice that it's not there. It's very difficult to understand, but if you think about your experience in the back of the head, that's something that you can imagine. You don't have anything to experience in the back of the head, and that's the reason why you also don't notice that you don't usually see, in the back of the head. However, if you have a particular part of the vision that can possibly experience something, and you are not experiencing in that particular location, let's say, if I cover my, right eye here, then I can't see it, some part of the region, right, far right here, which is not usually, visible. And if that is the case, then I can notice that, oh, I'm not seeing, and that there is abnormality here. So conscious experience is all about, in the end, the possibility of experience. And this is very important, which will come back in integrated information theory in week 10. And this is also important for psychological and also clinical kind of insight for how to rehabilitate a patient. Possibility of experience is quite important. Okay. So let's go to this case report. So the task that Gallant devised was a hue discrimination task. And then here the hue is very basic kind of, concept of the color. So color can be described in many different ways, but one of the ways to describe the input to the eye in terms of color corresponds to consists of the hue, saturation, and brightness, B, or sometimes called using nu or V here. And then when the brightness is low, then it becomes black, and if it's too high, then it becomes white. Okay, and the saturation means that the degree of the colorfulness starting from center to the periphery. Okay, and then the hue describes the variety of the different colors. So usually red is located in opposite side of the green, and the blue is opposite of yellow, and so on. And there's this coloring described like this way. And then the color hue discrimination task can be constructed such a way that maximum hue difference is 180 degrees. And normally we can distinguish the difference between the two colors, such as this orange and the yellow, or small shade of the green and blue, and so on. So it's a few degrees and so on. However, this patient, when tested with this type of the task, showed a dramatic performance degradation. So these UR, LL, and LR corresponds to the performance at the 3D opposite, unaffected visual field. So here, this is a UR, this is the LL, lower left, and this is lower right. And then there, when the hue is red as a baseline, they can detect the difference reliably at a few, 10 to 20 degrees of the difference in the hues. And that's not that different from the other patient under the certain, experimental conditions. Okay, and then this is the case when the green is used as a baseline. On the other hand, the upper left here is clearly, bad. So 100 degree means that if the baseline is red, when the color looks, was presented around here or there, yellow or blue, then patient can say that, oh, it looks different. Okay, so that's the kind of the deficit this patient has. And remarkably, when this patient was tested with some kind of object, let's say colorful patch here, moving from here to there to here, it seems like this patient will not perceive the fact that the color has gone from colorful to gray within this, and then again colorful. But again, as I said, if there is no possibility to experience the difference, then it's very hard to experience the difference, at least from the intrinsic viewpoint of the patient. So that's very interesting observation. Okay, so one of the controversy about the color and color qualia and V4 is that when the monkey physiologist has lesioned monkeys V4, then they don't really show the color deficits. It's very different from MT lesions and motion qualia. So it's been controversial. And so one of the breakthrough that came around the end of 20th century is the test for whether the V4 is really the NCC for color or not. And can you imagine what was one of the tests that they did? So it's a color after image. That's one of the technique that they can use. Okay, and then so what they did was to use the after image technique or after effects technique that they use for MT from last week, and then they applied it for the color case. So just a demo for this effect. I want you to fixate at center and then look at this screen. And that's the similar kind of after image demo like last week, but it's slightly different. So here I'm going to change blue and yellow, left and right, and then keep the red and green upper visual field. Then what happens is that after image of the blue and yellow cancels out each other, but the green and the red should be staying there. Okay, 3, 2, 1, 0. Okay, so for me it works really well and it continues there. Top left is blue and then top right is purple, and the bottom left and the bottom right is completely gray. So this type of the experiment can be utilized to look at whether the color after image is generated in before or not. So what happened? So Hajikani and colleagues did this experiment using fMRI in humans, and it turned out that all these color after image effect comes from not from B4 area here, and in fact it was found in B8, what they call the B8. So usually people term it, in the order of the discovery of the visual areas, and the B8 is probably the last one to be reported over the last 10 years or so. But B8 area seems to be the ones that reacts to color image. And here is this, color code for the strengths of the after image response. So what they did is once again, doing this alternating color time from 48 to 96 seconds, 48 seconds, like blue, yellow thing. And then when it turned off, then the areas, V8 areas response became weak, like immediately around the baseline. On the other hand, if the same duration of the exposure to the constant color was followed by the gray fixation, then there is a huge response at the offset of this, stimulus, corresponding to our own, color after image. And when they averaged all these, response and subtracted the fixation for these two situations, then they found that, V8 responds significantly at the onset of this color after image induction. So that's one of the evidence for the V8's involvement for the color experience. But also another version, another type of the evidence can be found from this type of microstimulation experiment. Here, the epilepsy patients who are, are inserted with lots of electrodes to locate their epilepsy location. And this is the small subset of the ventral surface of the brain. So this is the brain looking from below, and then upper part is eye and the frontal areas, and the back part is sort of B1 here, B1. And then the ventral surface of this right side and the left side of the brain is enlarged here. And then what they found was that the areas that were covered by these electrodes, which happen not to be corresponding to his location by epilepsy, generated the responses preferentially to the color image around these electrodes. And here, these electrodes preferentially responded to the face stimulus. And I'm going to come back to that later in this lecture. But anyway, so let's take a look at what happened to him when he was stimulated by this particular electrode 178 and so on. And a pair of the electrode 178 and next to each.
So he's now looking at a screen or sort of one of the meaningless object, and then see here at the bottom right, you see the microstimulation delivered into the color area of his brain, and that appears as a sort of artifacts of this EEG recording. So EEG recording within the brain is represented in a bunch of lines that goes from left to right, and each of the height corresponds to different channels of the recording. And some of them are color responsive brain electrodes, and some of them are not. But delivering electrical stimulation generates lots of artifacts. So here's the artifacts. And then he's nodding. And then it's terminated right now. And then he's going to describe his experience here. So he's actually a Japanese person, so you won't understand what he's saying, but the subtitle means is a good translation of what he's saying. Basically, he's saying that during this stimulation, left side of his vision becomes colorful. And also here, the same kind of stimulation, and then it's already terminated. And then looking at the face, and with respect to the nose, when he was looking at his left side starts to become like a rainbow color. Okay, and regardless of what kind of object he's looking at, it's always like the left side of the things that becomes colorful.
Okay, so in summary, this first part of the lecture showed that V4 or V8 is a likely candidate for the neural correlates of color qualia, and evidence comes from lesions after image in fMRI and electrical stimulation. And here again, the question comes to the same kind of thing as the MT situation. What is special about the neurons in V4 and V8 in generating color? And if you look at neuronal spiking behavior or biophysics or connection, it doesn't look like they are significantly different at the first look, but they are most likely different. And these local connectivity and local property of the neurons are likely to be the factor for generating color on one end and motion on the other end. But we currently do not have any theory, precise theory, except for most likely IIT integrated information theory like scenario, which we are also working on, is able to explain why it is that V1, V4, or V8 neurons can generate color while empty neurons can generate the motion. And this is, in fact, the hard problem of consciousness. Okay. So, but before going further, we will now go into the further evidence to look at the higher visual areas and its relation to the content-specific NCC in the next part of the video. All right. So next, we are going to look at the candidates for NCC in the higher visual areas. So by higher visual areas after B4, what I mean is mainly this ventral visual areas or inferior temporal cortex in the case of the monkeys. And also in the case of the humans, it is mainly about the fusiform face area and also parahippocampal face areas. This is a flattened version of the cortex again, and in terms of the flattened cortex, these inferior temporal cortex locates anterior to V4, and so these part corresponds to this IT areas, and it's consisted of at least in monkeys 6 areas at the time of this Christoph Scott's book publication, and it's more refined right now. And in humans, this corresponds to V8. Includes part of the posterior part of the IT around here. It corresponds to V8, the likely color areas for humans. So let's look at the case study where some subjects have received bilateral damage to this part of the areas.
Evan suffered brain injury after being hit by a car a few years back, and that caused him to lose the capacity to recognize everyday objects. We asked Kevin what he sees when he looks at things.
I see colors, shapes, punctuated by faces, the faces of people which I really recognize, and they stand out much more than anything else. That's why I can't read words or read music anymore. So I still can imagine what it looks like in my mind's eye, but I can't imagine what it looks like, or can't see what it looks like when I look at it. So essentially, I find things through color.
In fact, the brain needs both the structural and semantic processing to be able to interpret information transmitted through vision.
I can sometimes pick out small components of an object, and by using memory as a reference, infer what it might be. Like if something's long and silver, it could be a spoon or a fork or a knife, and usually I use my hands to touch the end of it, so I know, oh, it's a fork, or it's a knife, or it's a spoon. So memory does play an important role in that, and in everything I look. I mean, I come to this chair. I know it's a chair. I don't know what type of chair it is. I know it's pink, but I was able to sit down in it. But this is my house, so I know where everything is. Before I could read music, but now I can't read music anymore. So I deconstruct music in my mind's ear. So I listen to the piece and I try to separate the different instruments, what they're playing, so the bass part, the treble part, the melody, and then or any harmonies, and then I reconstruct it back for the guitar. So I don't actually, I'm not listening to something, a singularity is broken down. I think we do that when we learn about anything. When you're a child and you take something apart, you deconstruct it to find out how it works and you put it back together again. And then you have this intimate knowledge about how the object works and what it is. And I do that with in my mind with things that I come across, especially with music anyway, deconstruction. That's my son, my oldest son was Santa Claus. And this is one of my youngest son. When he was younger, he's a teenager now. And, yeah, that behind that angelic face is a demon child. That's my eldest son when he was about 6 weeks old, 2 months old. I think that's me here. And that's Hayden, my youngest son, when he was a baby.
And do you see the details of the face? Do you see the eyes clearly? What is it in the image that you recognize?
The whole face. And I'll tell you why you can say I can see a whole face, but I can't see a whole object. Like this thing here, I only see parts of it. Like I see these diagonal things here. I can't see the whole thing. But with the face, I see the whole thing. So you say, well, how come I can't see that in its entirety? But yet I can see the face in its entirety. It's because a completely different center of the brain processes faces and processes objects. And in me, the part that processes objects, symbols, has been damaged. But the part that processes faces is fine. And that's how I'm able to recognize people's faces. So I recognize my colleagues at work, my family. That's much, I'd much rather have the condition I have. I'm lucky. Because I'd rather have that than not be able to recognize faces. That would be awful. Not being able to see the people you love.
Okay, so that was the case report and the subject reports from the patient Kevin, who got agnosia of the object. Agnosia means that lack of the recognition despite of intact parts of the object. So in his case, he seemed to have quite a good understanding of the neuroscience, and he talks about the fact that he can recognize faces, but not the VAS, for example, is because he has a different areas of the brain specialized for face processing and object processing, and that's probably likely to be the case in his case. His case is probably the lesion specifically on the object areas, but not the face area. So by the face area, what I mean is this area called Fusiform face area. And that's sort of a ventral surface, and it's bigger on the right side. By the way, again, right and left is flipped here. Usually on the right side, there is a bigger Fusiform face area in the people. In my case, when I did the experiment by myself, I found right side is much bigger. And then the PPA is a parahippocampal place area. So the parahippocampus meaning that it's closer to coming to the hippocampus, and it's more medial, the center, compared to the face area. And it is specifically responding to the image of the face houses and also landscape and the outside scene and so on. This is areas that are discovered around 1997 or so by many different kind of the neuroimaging study established around that time.
So here, this is a typical fMRI experiment of roughly like 6 to 5 minutes, alternating between the fixation cross, just staring at the blank screen, followed by 16 seconds or something like that of faces with different example of the faces. And then again, going back to the fixation, and then next one is the house, repeated for many different kinds of houses and so on, and then face, house, repeated within the same subject. And then, as you can see, there are some specific areas of the brain, especially around Fusiform area, that increase the activity during the face perception, and the house, another area that responds to house, many different kind of house. And then typically subtraction between the two, or subtraction between each of the conditions to the fixation period, gives a functional definition of FFA or PPA for each individual subject. So this Fusiform gyrus is a term for the anatomical area, or parahippocampal gyrus is anatomical areas, but a face area or place area is a functionally defined areas, and each person has a slightly different location within Fusiform or parahippocampal gyrus. Okay. So using this technique called the functional localizer, now people can routinely localize where is your FFA, where is my PPA, in 6 to at most 10 minutes or something, and then that area tends to reliably respond to any kind of faces and any kind of places or landmarks or houses. And this is very useful because it can, with this, you can isolate areas based on the functions, and then test various hypotheses about a particular category of the stimuli, such as face or a place and so on. And as you will see, this has been used for addressing the question of the consciousness and attention. All right. So, in the case of the previous patient, Japanese patient who was implanted with the face and the face area and the color area, here, the color areas were more posterior, but the face area was more anterior. Okay, and the extent of the face areas are unclear because we don't have the electrodes across all the entire visual cortex, but ventral cortex. But now within this electrode placement, this is part of the face areas. And these figures on the right side, by the way, is based on the responsivity of each electrodes. Of course, tested with a face image, house image, color image, and so on. And then the size of the ball shows the strength of the response in the log of the p-values. P-values of 60 or 15 is a very gigantic, significance, and it's almost like if you do the prediction of which categories he was seeing, it's almost gives you 100% accuracy based on this, areas of response. But now the critical question for this is what happens if you stimulate these FFA areas? And here, pair 181, 182. So that corresponds to this area, this pair. If you stimulate, what happens? That's the question. And then, just to remind you, 177, 178 pair here. This was a pair that was stimulated when I showed the video. And he was staring at the box, and it says that, oh, left side of the box looks like a rainbow. And then when he was looking at the face, he said that if I look at the face, this side will look like a rainbow and glowing on the left side because it's a right hemisphere. Okay. And it's important to note that these electrical stimulation is controlled by the doctor, and the patients are never possible to guess which part of the brain was actually stimulated. So all these bandage is connecting to the electrodes, and then all the wires go behind his back, and then goes to the stimulation computer that is located in the other part of the room. And then doctors who control this stimulation electrode is invisible to him. And so, he also doesn't feel anything in terms of pain or tingling sensation when he received any micro stimulation for these areas. So given that this is very important kind of observation, okay, because it's a pure double-blind kind of experiment. And then when he was stimulated with the face electrode, and then when he was seeing the face of this doctor, then he said, "Oh, your face completely changed. I don't know what's going on. Your eyes changed." That's a typical kind of description of the face area stimulation. Okay, and then also when he was stimulated while he was looking at this book's face, then he says that just for the very first second, I saw an eye and the mouth. So in this case, this particular stimulation seems to have caused a part of the face, like impression on the object. And this paper describes other types of the objects, and he seems to spontaneously talk about rainbow color or the face upon the stimulation. And so this is very compelling evidence that an FFA is sufficient to generate a face conscious perception or a face NCC. And after these experiments, not only these, so not only these experimental evidence in humans, we also know that monkeys actually do also possess the face processing system. And this is a coronal slice of the monkey in fMRI, and done in 2006 or so, where people started to record the neural activity while or insert the electrode based on the fMRI or MRI guidance. So this is a experiment where they first did this face localizer for monkeys. So bunch of faces, bunch of houses, bunch of flowers, and so on. And then found this particular patch in this inferior temporal cortex contain the faces. Okay, so these yellow things are the box cells that responded to this faces much more strongly than any other categories. And then note that very close to this is an auditory cortex. And the monkey's brain is much smaller than humans. It's about the size of this fist. Also, so even though it looks like big, in fact it's very tiny. And you can imagine that recording this area or missing just by 1 millimeter, you don't find any face patches. Okay, but what this Doris Stahl's group did with this monkey fMRI combined electrophysiology is that after localizing face patches and then target the electrodes into that area, then they found that 90% of the visually responsive neurons over there are strongly face selected. So this figure shows a summary of many different kind of the images, like 16 faces, 16 bodies, fruit, gadgets, hands, and then scrambled, and then looked at the firing rate of these neurons. And really surprisingly, this is across 180 cells, but almost all of them responded any kind of faces, but not other. So this entire patch is almost purely composed of the face responsive neurons. And by the way, one of these important finding for the face neurons in IT is that they tend to have a huge receptive field compared to the B1 or B2. So as I mentioned in a couple of weeks ago, neurons in the lower visual areas tend to have a small receptive field, and the smallest in the retinal ganglion cells. And then as you go from retinal ganglion cell to LGN to B1, it becomes slightly bigger. and then B2 even farther bigger, and B4, and then IT covers almost like sometimes almost entire visual field. And what it means is that when you are looking at the firing rate of these neurons, then faces that are presented in anywhere in the visual field generates the response to these neurons. And that's the sort of idea of the receptive fields of the IT neurons. And this is very important considering whether they are the neural correlates of consciousness, okay? So, and this is example mentioned in the Christoph Koch's book, but also not only the faces, whenever monkeys get trained to be familiarized with some kind of target object, let's say in this case paper clip, bended paper clip, and then monkeys were trained to discriminate its orientation for several months. Then they found some neurons that respond to this particular object of a particular view in 3D. So in this case, monkeys are basically understanding that these clips are different view of the same electrodes, same paper clip, and this is different, bending, and this particular view generated a huge response of one particular IT neurons. And this type of the very selective complex response property with the huge receptive field is a hallmark of the IT neurons. And this is another types of the face neuron. So as I mentioned, Dorstal's experiment was conducted in terms of follow-up of all these experiments. But previously, people found that if you put the electrodes around there, sometimes you find the neurons that respond exclusively to the particular face. In this case, it is the face of, I think, David Leopold, which I'm going to talk about towards the end of this week's lecture. And in particular, in this particular neuron, responded more strongly to the profile of the side view of this face. And slightly different angles or opposite face, angle this side versus this side generates a response, but not to the front or back side. And it's also specific to this beard face, but not other faces. Like this one, it didn't, and this one somewhat smaller. And then these Rubin's face illusion, it didn't respond much. Interestingly, monkey, when they saw the face in this face versus illusion, they produce much stronger response. But when the contour is such that we see VAS, monkey also seems to see VAS, and that's corresponding to low firing rate. And these four is a low resolution image, so it's a bit difficult to see, but it's all monkey faces, and that doesn't also respond, generate lots of responses. So these specific and also rotation specific neurons are found in IT in monkeys. And this is one of the Nature papers from Christoph Koch's lab. And then when they were publishing this paper, I was there as a grad student, and they were quite excited about this finding. So what this is figure showing is another types of experiments recording the single neuron response from the epilepsy patient. And epilepsy patients, you saw the video a couple of minutes ago. They are implanted with these electrodes to localize the four sides of epilepsy. And we are going to talk about epilepsy loss of consciousness in week 11 or 10. But for those people, because this epilepsy is so devastating, they need to have an first try the drugs to soothe the seizure. However, in many cases, the currently available drugs have either side effects or not really useful. So some of the patients resort to this surgical resection of the seizure foci. And to do that, minimally remove this seizure foci, they implant the electrodes and monitor where the seizure originates from. And if you don't do that, then you might cut too much, and then you might have heard of this very unfortunate person, HM, who was taken a lot of cortical areas involved in the memory, and then he lost memory for his life. But anyway, so this is the medial temporal lobe that includes hippocampus and also amygdala and the entorhinal cortex and so on, which is typically regarded as a high-level recognition or cognition memory, is the target of this epilepsy recording. And here, what they found was that so they looked at the neuronal firing property of this recording from the hippocampus here. Okay, and then based on the previous experiment, they kind of expected that the hippocampal neuron has an preference to the images that patients are familiar with. So as I mentioned, after practice of the bending, clip and so on, monkeys tend to show some specific, response to the bending clip in his IT, and the hippocampus is receiving the input from the IT and so on. So what they, Quiroga and so on, did was to do this screening procedure. So in, let's say, day 1 or day 2, they would ask the patient what kind of movie they watch or TV series, and also what kind of hobby the patient has and so on. And then in this particular patient, this person really liked watching the TV show Friends, and also he or she watched she really liked the basketball and various different kind of movies, and she wanted to go to Australia, so that's Sydney Opera House, or Paris, the Pisa, and many other areas. And she also reported watching, hating or liking spider or snakes and so on. And then they collected all these images from the internet and then showed each image one by one to the patient. And then after several weeks or several months of the analysis, they found all these responses. And then they also speeded up all these procedures so that now they can do it in just like 30 minutes or so during the lunchtime. And then after finding this particular neurons that found okay, so this neuron seems to be responding to, let's say, one particular image, then let's get many different kind of examples that would be categorized with this type of image during the lunchtime, and then present the patient again in the afternoon. So that's a kind of the really long story of this behind this experiment. But what's in the end what they found was that in this particular one neuron that fires this face photo of the Jennifer Aniston, okay, and then each of the row corresponds to each trial presentation of 1 second of the Jennifer Aniston. Okay, and all the subject was doing is that pressing the button if they saw the face, and then if they saw non-face like this snake, they don't press the button or something like that. And then because they found that this particular neuron seems to respond to Jennifer Aniston, they collected lots of different versions of the Jennifer Aniston photos. Okay, and then presented him. Then what they found was that this neuron really responds to any kind of Jennifer Aniston. This is 5 or 6 trials, and then this is an average firing rate. And notice that difference of the Jennifer Aniston photo is quite substantial. Background or color or emotion is very different. And also interestingly, this is still potentially anecdotal, but when they paired with a Brad Pitt, Jennifer Aniston with Brad Pitt, you may not know, but they were married at the time. Then the response became very, very weak. And this may be because the neurons are not really, not really predicting their divorce in the end, but Brad Pitt neuron, which may, exist nearby that neuron, may be inhibiting this Jennifer Aniston neuron, or maybe because of the attentional selection going on, and then when they are paired with the Brad Pitt, patient may attend to the Brad Pitt more, or something like that. We don't know, but what we know is that the response to the Jennifer Aniston with Brad Pitt became really low, and all other neuron response, for different types of the female actors, and also basketball or any other stuff, it didn't respond. But this was very specific, but specific in terms of category of the Jennifer Aniston. But as long as it's Jennifer Aniston, then it responded. Here's another example. This is a right anterior hippocampus. And then in this case, this neuron responded to Halle-Valley theory. And this is again, found within, the screening session. Oh, this one particularly responded to Halle-Valley. So they've tried to find many different kind of images by Halle-Valley, and but they couldn't find many images like this. So they also presented a line drawing or a cat woman. Okay, so this was a part of the Batman series, and Halle-Berry played in that movie as well. And also this is the text of the Halle-Berry, and the other different kind of text was also tried for this particular neuron. And then what they found was, again, surprisingly, not only these images of Halle-Berry, but also a drawing, and also even cat woman generated a response, and somewhat bigger response was also produced by the text of the Halle-Berry. Okay, this is again pointing to the fact that this neuron may be related to the conceptual understanding of the Halle-Berry, but not the visual property or scene of the Halle-Berry itself. But this is a kind of a response that we can find if you have a chance to record the neurons from the humans in the hippocampus. And then one more example. Here is the neurons that responded to Sydney Opera House here. But also this Buhari Temple. I think this is this. This is also quite interesting because this person didn't anecdotally differentiate these two types of the buildings, actually, Buhari Temple and Sydney Opera, at least, seen for 1 second, looked same to this patient. And that generated this response. But because of the difference in, shape, maybe that's the reason why overall response looks a bit different. But of course, it doesn't respond to other types of the images, maybe a little bit for this one, but many of the buildings, famous buildings, Eiffel Tower, Pisa, Tower of the Pisa doesn't generate a response. So in terms of the search for the NCC, what's also important to do is to do this type of the experiment with the NCC paradigm, right? So one of the experiments they did was this backward masking experiment. After identifying the neuron which responds to this World Trade Center, they presented this with only very short time, 33 milliseconds up to 264 milliseconds, and then followed by the backward maskers. And then when the subject recognize the image, they need to say, so here the red one corresponds to the trials. Subject said, oh, I don't understand it. Okay, and sometimes when it's a face, for example, Mother Teresa or somebody else, then the blue trial exists. And these are the trials when subjects saw, oh, I saw the face, or I saw the Mother Teresa, or something like that. And then in any case, at 33 milliseconds, there was no response. But at 66 milliseconds, when subject responded or saw the word through the center, then they showed the response, but not when subject denied seeing. And same goes for 132 or 264 milliseconds. So here, the stimulus is the same in this, three cases, but the conscious reports are different between blue trial versus red trial. And only blue trials of seeing World Trade Center elicited a response. Okay, so this is again consistent with the interpretation that it is a conscious recognition of the or concept or understanding of World Trade Center that evoked this response of this particular neuron in the right entorhinal cortex in this case. And this is a population response across many different neurons recorded from the same similar areas, and showing that the separation of the trials based on the visibility, either recognized or not recognized, within each of the duration, so seems to differentiate the response. Okay. It's up to if it's let's say if we look at the threshold trials around here, when patient recognize the image, then it's about 1.5 up to firing rate. This is normalized firing rate, but it doesn't reach up to 1.5 for the unrecognized trial. But it still increases firing rate. So this amount here may be corresponding to unconscious response of the neurons, or it is generating something, but it doesn't result in the reports in the patient. That's one interpretation. So in summary, in this part of the lecture, what I talked was that the inferior temporal cortex and the ventral temporal cortex, including the medial temporal cortex, is really crucial for object or face recognition. And the lesion of these areas can result in object agnosia, like Kevin's case. And if it's in FFA face area, then prosopagnosia can result. And fMRI activation can be used to identify FFA and PPA functionally, and also stimulation of these areas can generate a face percept. And the PPA can also is known to generate some kind of topographical percept. And also the targeted recording within the face patch in the monkey reveals concentration of the face neurons. And also similar kind of effort can be made for human medial temporal recording, lobe recording, and then find even more finer and also specific type of the responses can be found in the medial temporal lobe. And one of the things that I wanted to point out is that localizing the areas first, and then combine that with the stimulation or report of the phenomenology from patients. And then also, if we have an opportunity to do the recording of the neural activity, this constitutes a really powerful method for the future NCC. And just without doing any fancy experiment, it is still already quite striking and many things we can learn. And this is still like there are lots of things to be done in this area, but this constitutes a very promising future research. So what does it mean? So monkey IT and human medial temporal lobes neurons are very selective in terms of response property, and to the extent that it only responds to Jennifer Aniston and not with Brad Pitt, right? But you can ask yourself, do they correspond to what we report or what we see? That's the question for the consciousness, actually. Also, as I mentioned, these neurons tend to have a really big receptive field. So the response itself may not differ when the face is here or there or there or there. Also, I didn't strongly emphasize, but
the neurons in a medial temporal lobe is very, very sparse in firing. It usually fires only a couple of spikes. Here, for example, over 1 second. So 1 second is from here. So in the case of the Jennifer Aniston, the response from here to here is 1 second. But these neuron spikes only once to 5 times. Okay, and it's really transient. And yet, our experience of the Jennifer Aniston stays, or at least stretch from the time we see it, and then to understand that, oh, it is hard, until the end of this presentation. So in terms of also temporal duration, it seems it's not that straightforward to regard these type of response as the neural correlates of consciousness of seeing Jennifer Aniston. So this also prompts us to think about, let us think about, whether the single neurons can be taken as any kind of, response mechanism for conscious percept. And if not, which probably many people already shares an intuition, maybe some kind of population or areas of the neurons are necessary to generate a conscious percept. But what exactly is it? That's the question. So to address this kind of question, the next question is about whether is there any better paradigm to specifically talk about NCC search, and that's the binocular rivalry we are going to talk next. Okay, so this part is about introducing the binocular rivalry as a potent tool for the NCC research. And this is the schematic from Chris Fritz's paper in 1999, and depicting as binocular rivalry as a paradigm situation of the perception. Subjective experience changes without the stimulation, and the behavior remains constant. And here, the behavior remains constant part is a bit problematic because people can report the percept. But binocular rivalry's case, you don't need to report, and then still experience changes without stimulation. So that's already forcing what's called a no report paradigm in a sense. So here's the different ways to set up the binocular rivalry experiments. The most easy kind of setup, if we were to do this lecture not online but in the lecture hall, I would actually give this red-green filter to each of you, and then let you see this image, for example. Then through the red filter, you see only face, and then through the green filter, you see the house. And then the percept of the face and the house alternates between them. That's one typical binocular rivalry experiment. And also this can be also done by using 4 sets of the mirror like this way, and then see completely different image without any kind of leakage of the color or any kind of constraint, to see completely different image on the left eye and the right eye. That's also another typical set. And then in fMRI, you can also do this kind of thing without introducing mirrors or filter by having a prism, and then bend the light direction just in front of you, and then see different images through this kind of occluder. This is like sort of the glass in front of you, but pointing into different direction. And so it's relatively easy and cheap to do. So, but binocular rivalry also doesn't require any kind of expensive setup. So this is, for example, binocular rivalry in everyday life. in this case, what you need to do is the following. So first, you put the finger in front of you, and then let's say around 15 centimeter or so around here, and then look to the sort of the in front of your display or mirror or something like that, and then pay attention to this finger. And then what you'll notice is that the binocular rivalry is actually happening at the border of the fingers, and then you see sort of the by adjusting the size, distance between the finger, you can have a sort of the floating sausage in between the finger. And then also important thing to notice is that if you don't move your eyes or blink and then focus for a while, the finger starts to get attached to the original finger or floating, or two finger actually also becomes one. So there are three different kind of phenomenology. One is most typical, the floating sausage and two short finger. And then another is completely connected fingers. And then other two is one finger connected to the central floating sausage and so on. That's one binocular rivalry experience in the everyday life. And then another one is this demonstration using the paper. So, for example, if you have any kind of paper in front of you, I strongly encourage you to try this and also show it to your friends or family. So here you have a paper, and then you just roll it up like this. And if it's too long, it doesn't work really well. But something like this, that's fine. And then hold it at the end like this, okay? And then look at this. Hold it with the left hand, and then put this hold to your right eye. And then if you open your eyes, both of your eyes at the same time, then you should see that with your left eye, you should be able to see your hand. But with your right eye, you see only hole through this, column, and then that should coincide roughly to the hole in the face, hand. And you can do something like this to make the hand more, dramatic, and or any kind of, combination of things, angle like this. For example, I don't see, my hand now becomes very thin. There's a hole here. And again, you can notice that if you don't move your eyes or blink and then wait for a while, then the hole gets sort of closed, and then again opening up, close, and then open up. And then now closing, up. And this happens at the time period that is sort of peculiar for each person. It's not deterministic. Okay. And then this type of the binocular rivalry also can also happen in front of the mirror. So this is the relatively recent kind of finding that we made and reported to the science community last year, and then published this year. So this is we call the third eye rivalry. To perceive this, you need to go in front of the mirror, and then just like standing 30 to 50 centimeters from the mirror. And then what you're gonna do is to look at your right eye with your left eye, and then with your right eye to look at your left eye. So it's like a crossing eye a little bit. And then if you can achieve the fixation on your one of your eye with both of your eyes, then you can achieve some kind of situation where your face is composed of 3 eyes plus 2 faces. And then because the eye has a very distinct contrast, you can
maintain the fixation relatively easily, I think, unlike other cross eye or parallel eye visual illusions. And then if you wait for a while, then you'll see that only one face dominates, and then the other side of the face becomes kind of a background or invisible, or the other way around. And then what I often also see is this only one eye cyclopean kind of situation without seeing the faces. And this is also the binocular rivalry in everyday life. And the fact that this was published only this year means that many of this type of the binocular rivalry in everyday life hasn't been reported. And probably if you find some, you may be able to publish as a paper. All right, so what this means, at least three of these examples, is that the binocular rivalry happens as a sort of the even not only in this kind of artificial experimental situation, but also everyday life with the fingers or holes, or in front of the mirror. And what is causing this binocular rivalry is essentially the discrepant images projected onto your right eye and the left eye retina. And then our conscious experience has this property where on one location, you tend to see only one object at a time. Therefore, that's one of them is perceived one at a time. But it doesn't. The brain doesn't commit to one interpretation all the time. It switches the other ones, and that can be explained in many different ways. And psychologists have been discussing about the mechanism for a really long time. But we are going to talk about the neural correlates of consciousness using this binocular rivalry. That's the plan. Okay. So, but having said that, first important kind of thing to realize is that there are lots of psychophysics experiments done on the binocular rivalry, and we know quite a bit about the property of the binocular rivalry, from individual difference to the property of the rhythm, and also the property of the images that causes that. So, for example, the left side is a typical histogram of the duration of the binocular rivalry. This is again from one of our papers that let's say in this particular situation of the binocular rivalry, then the switch between the left eye to right eye happens most frequently with a frequency duration of 2.5 seconds or something like that. So you see one image with the right eye for 2.5 and then another one for 2.5. And sometimes it's very short, like only a fraction of a second. And then sometimes it can be really long, right? 10 seconds or 15 seconds. It's quite remarkable that if you try this or third eye rivalry or this column thing, that you feel like you can control the duration of binocular rivalry, but if you control many things very carefully, especially blink and eye movements, it turns out that you can't really control binocular rivalry. And the rivalry happens mostly involuntary and with much of the effects of attention almost like minimum compared to other types of the binocular, the bistable illusion. So, so this type of distribution is called the gamma distribution, and that's one of the binocular rivalry's property. Okay, and then the other one is that contrast dependency. So here, for example, if I present the left eye 100% contrast grating, and then the right eye is horizontal grating, and then this proportion of the contrast could be like X%, and the X goes from 0.3 to 1.0. Okay, so this is a right eye contrast in this situation. And then it's very counterintuitive in the beginning, but what it means is that as you make the strength of this thing, from really faint, kind of horizontal, to very, very strong horizontal gradient, what happens is not the dominance duration of this grating, to change a lot. So what it means is that even if it's very weak, it dominates roughly 2 seconds on average. And then even if very, very strong, 100% contrast, it still dominates only like 2.5 seconds. So the peak here, or mean, doesn't change as a function of the strength of the contrast of the stimulus itself. However, what changes is that if this one becomes weaker and weaker, then left eye grating, okay, in this case it's a vertical grating, that becomes really long in dominance. So each time it becomes visible, it gets visible for, let's say, on average 8 seconds. And then come back to right eye, but it's only, dominant for 2 seconds, and so on. And then as the duration of the strength of the right eye becomes stronger and stronger, and then to the extent that it becomes now the same, then the dominance duration becomes more or less the same, left and right eye. In this particular subjects or experiment, left eye still had some kind of dominance, but mostly this becomes same when the contrast becomes the same. This is a very peculiar property, and not so many people don't know about this. And so this can be used as a sort of a check of whether the subject is doing the task properly or not through the experimental analysis of this test. And this can be applied to animals or patients or kids or whatever, okay? So this is important. All right. So now also, I wanted to introduce the technique that I discovered when I was a graduate student. So here, in fact, the reason why I discovered this illusion was because I was translating chapter 16 of the Christoph Koch's book at the middle of the night, like 2 a.m. or 1 a.m. or something like that. And then if you read that chapter, you will encounter that the discussion about binocular rivalry and so flash suppression. So flash suppression is similar to binocular rivalry, but you present the stimulus to left eye and then introduce the second stimulus to the right eye later. And then if you do that, right eye stimulus tends to become dominant. And usually up until that time, flash suppression and binocular rivalry was kind of treated as related but distinct phenomena. And then while I was translating that chapter, I just came up with this idea of, okay, so if I intermix binocular rivalry and the flash suppression, what happens? And then I went back to the laboratory in the midnight and then programmed this stimulus. And then what I found was that this, the other stimulus that kept on sort of doing the flash suppression dominated for really, really long time. And at that time, I couldn't see the suppressed stimulus for a long time. So I even doubted maybe the bug of the program. But it turned out that it is a discovery of this situation of continuous flash suppression. So basically, what it means is that here one side of the eye receives a stationary image. Let's say even the angry face or something like that. And that was considered at that time very, very difficult to suppress in a binocular rivalry. The face is super salient and the emotional stimulus makes it also very difficult to suppress it. However, when I flashed the other image to the other eye, at roughly the period of 100 milliseconds for one image, and then I don't repeat the same image. So here I'm using like, let's say 64 patterns of the different, Mondrian, like, abstract, images. Then most likely because this image is effective to stimulate the binocular, the neurons in visual cortex throughout the visual cortex B1 to B2 and so on, because it has a lots of edges. That is a very effective stimulus to activate various neurons, and also because I don't let them adapt for each of the neuron. It responds to the onset, but the next one comes 100 milliseconds after, and different neurons are evoked by that next flash. And because this one, in a sense, avoid any adaptation, as you saw experienced last week, it is very effective in driving the other eye for a long time. And then as a result, the percept is dominated by this flashing stimulus. And this is very, it turns out to be a very potent stimulus. And then it allows us to study the non-conscious processing of the brain quite effectively. And some of the insights about consciousness and attention comes from the result of this continuous flash suppression technique from next week. Okay. And some of your tutorial presentation depended on the continuous flash suppression, CFS. Now, just a quick sort of, explanation of what is a potential neuronal models or binocular rivalry or CFS. So here, the key finding is this thing. That strength of the one eye's stimulus does not change the dominance duration, but the other eye's dominance. Okay, so that's the key component to explain and also expect what types of the neural mechanisms are responsible for binocular rivalry and also CFS. So here, the retina and the LGN and up to V1 is expected to more or less, generate a retinotopic response to the left eye and the right eye. And then within V1, at least layer 4, the neurons are responding to only input from left eye or right eye. And then even though after that, it becomes sort of merging to binocular neurons, there is always a preference from left eye or right eye, and so on. And then one important insight is that the same orientation preferring neurons are likely to be exciting each other. Okay, that's a red arrow. And the different orientations are inhibiting each other. So this is a very nice kind of situation to expect how this stereo image or usual binocular situation to arise, right? Left eye and the right eye tend to see something very similar, and if the left eye input and the right eye input is the same, then they tend to facilitate each other. However, unlike the normal situation, if there is an binocular incongruence, so left eye is receiving this vertical and the right eye is receiving this horizontal, then they are trying to inhibit each other. Okay, right eye is saying that it should be horizontal, left eye is saying that it should be vertical, and this inhibition is making somewhat, once this guy right eye starts to win, then that starts to inhibit left, so it becomes it facilitates a clear winner. But this guy right eye preferring neuron starts to adapt, and its response to goes down, and then that allows this left eye neuron to become dominant, and then now the switch happens, and this continues on and on, and that's the binocular rivalry neural modeling. And also, important thing is that here, the effects of one's activation is mainly on the other eye's neurons through this inhibition. And that's the reason why one stimulus is strong, as the neurons, let's say, as an input to the right eye becomes stronger, it doesn't have a sort of the sustained input to make it more sort of the dominant, but the duration of the other eye becomes shorter through the strengthening of this inhibition, and vice versa.
So that's the sort of the minimum model of the binocular rivalry. And still, there are lots of remaining questions about binocular rivalry, and this relates to also somewhat relating to the neural correlates of consciousness. Okay, so for example, what is the mechanism? Neural mechanism of the suppression is still unclear, and what is the fate of the suppressed stimulus? So suppressed stimulus becomes consciously unavailable, invisible, but can it actually affect our behavior? So that's sort of the question about non-conscious processing, scope and limits of the non-conscious processing. And the third is how does the nature of the stimuli affect binocular rivalry or CFS? As I mentioned, in the case of the binocular rivalry, angry stimulus, for example, is very difficult to completely suppress, but CFS can suppress it. And CFS utilizes these Mondrian-like patterns, and that prolongs this binocular suppression. So there are lots of questions about what kind of situation or stimuli achieves the best suppression or weaker suppression and so on. That's sort of another question. Also, another question would be that what is the neural correlates of the visible and the invisible stimuli? It's sort of related to number 2, but also what is the neural activity that corresponds to visible and invisible stimuli? And then finally, what is the neural mechanism of the perceptual switches? This also becomes important under the no report paradigm discussion in 2 weeks from now. So in sum, in this section, I introduced the binocular rivalry, and it happens in everyday life, and under the constant stimulation of binocular rivalry, conscious percept flips between 2 alternatives in a random manner, and CFS can suppress the stimulus in a more reliable and longer duration. But a precise mechanism of binocular rivalry or CFS are still unknown. And what is the neural activity during binocular rivalry is the one that we address in the next video. Okay, so this is the last part of the this week, and if it gets a bit longer, then I'll make sure that next week is going to be roughly around 90 to 100 minutes. So be sure to be balanced. Okay, so to do the recording of the neural activity during binocular rivalry, it's best to do it in monkey subjects. But the problem of this monkey experiment is, of course, to make sure that monkeys are actually experiencing something very similar to humans. And in the course of establishing this binocular rivalry in monkey, there were lots of breakthroughs in terms of the neural correlates of consciousness, and that's why I'm going to go into a little bit deeper into this particular experiment. So first, to do this type of the experiment, monkey physiologist has to train the monkeys to so that they can report binocular rivalry genuinely, and that's already quite a bit of the task. And it is actually indeed possible through like Reva press and so reward and so on. But also important thing to notice is that the monkeys are also like humans to some extent, that they can actually reward do the task without much reward if it's kind of interesting task and intrinsically rewarding. So that's one of the things I want to mention. But having said that, this training of the monkey for binocular rivalry takes years to do. And once they apparently understand the task demand, like reporting this in certain way, then another thing to make sure is that whether the monkey's psychophysics behavior report pattern is similar to humans. And then to do that, we need to collect the binocular rivalry behavior data in monkeys. Okay. So the first thing these people did in this Nature paper was to convince the researchers that monkeys are actually experiencing something very similar to humans. And important variables are here for this gamma distribution of the phase transition. After seeing this red or horizontal from right eye, and then there is no prediction possible from right eye duration to the left eye duration, and also the duration is distributed like a gamma function, like here. And in fact, so this is typical human observers, and monkeys also exhibit very similar kind of a gamma distribution pattern. Okay, and that's one of the evidence. The other evidence, which is much more convincing to us, is that under the same equipment and the same stimulus, humans exhibit this contrast, highly counterintuitive, contrast-dependent, mean dominance duration. And the monkey also did more or less reported the same way. Even though there is a crossing here, it's just based on the right eye, left eye, dominance difference for this particular human and particular monkey. So, in essence, monkeys and humans show very similar contrast-dependent modulation of the binocular rivalry. Strengthening one eye's input changes the duration of the other eye, which is a hallmark of the binocular rivalry psychophysics. Okay, so that was very convincing to the researcher. And then the next step was to try to train the monkey to fixate at the small points of the display and train them to hold it for a really long time. And this turns out to be extremely challenging. And usually, monkeys and even the humans cannot hold this kind of thing, the gaze for a long time. But the researchers for this experiment was able to train the monkeys to hold a fixation for a long time within this, roughly 0.1 degree of the eye position. And remember, 0.1 degree is much, 10 times smaller than this finger's thumbnail in front of you here at arm's length. This, remember that there is a roughly 1 degree of visual angle, and 10 times smaller is the required fixation window for the monkey. And this heroic monkey was able to do this for a long time. And during that period, then researchers were able to present the binocular rivalry stimulus so that that covers the monkey, the recorded neurons receptive field completely, and then presented to one eye, one stimulus that the neuron prefers. For example, in the case of the B1, as I mentioned a couple of weeks ago, B1 neurons can prefer the some direction of the gratings and also orientation of the grating with certain direction of motion. And the MT, even more so. But B1, B2, B4 neurons also have each particular orientation preference and also direction preference. And then pairing with that, 90-degree different orientation gratings will be presented to the other eye. Okay, that's a sort of setup of the binocular rivalry experiment. And then here's one such recordings trace. So just to go slowly here. So what I'm showing here is that roughly 20 seconds of the data from one trial of the monkeys. Okay, and the monkey was holding the levers on the left side and right side, and then left lever to push or pull to report that it is seeing the left tilted grating, and the right lever to report that it is the other side of the grating. And the monkeys were presented with this conflicting grating to left and right eye, and then that's indicated by this one. Okay, so from time 1 up to time 13 or so, monkeys were presented with this binocular rivalry. And then the next thing is the eye movement wise. If it's blue, that means that monkeys were fixating within this 0.1 degree of fixation. So what the neurons are responding to is precisely on its receptive field. So you can basically ignore the data that coincides when this part becomes black. Okay, so this part black means that it's a bad fixation. And then also another thing is a perceptual report wise, when the monkey says that it is seeing this right orientation like this one, they are playing the lever for this red here, and if it's green, it's the other side. Okay, so in this particular trial, monkey started to pull the lever to indicate it's a left orientation. Okay, and then here right orientation, and here left, and then right, and then left, and then right, and then left, and then here the unbeknownst to the monkey, researchers changed the stimulus to right orientation, and then 1 or 2 seconds later, monkey started to press a button, and then that's the end of the trial. Okay, and then this vertical tick mark corresponds to the neurons one spike, and in behind this tick mark there is this smooth line that corresponds to the firing rate of the neurons in an instantaneous manner. Okay, so that's all about this figure. And then from this, what we can see is, first of all, at the onset of the stimulus of the binocular rivalry, there's a huge response. And this is quite common across all the neurons. They tend to respond to the onset of the stimulus, and so we ignore. And then after that, every time when the neuron, the monkey responds to press right orientation here, from left to right, okay, then there is a period of neurons firing around here and then here, and then after that there is also a firing here. And this corresponds, in this particular case of the neuron, is the preferred orientation of the grating for this particular neuron. So it makes kind of sense. Remember that in binocular rivalry, if you do it with this finger version or face version or whole, there is a transition period. And when monkeys pull or push the lever, there is always unclear period where the neurons, the phenomenology is starting to gradually change from left to the right and the right to left. So it's understandable that if it's starting to fire before the perceptual switch. Okay, so this is a particular trial. And then next, so they recorded this type of the trial for really a lot. And this is still like one particular neurons experiment. And while recording from this neuron, and they did lots of trials. Trial 1, trial 2, 3, and so on. And then what I said more or less holds true, right? So in the beginning, there's an indiscriminable response. And then whenever there's a response to report on the left tilted orientation after a while, then neuron tends to fire more, and then there is some kind of delay in firing. On the other hand, if the neurons or if the monkey started to report seeing the other orientation, it tends to be preceded by not so much of the firing, but here this is the opposite. And then here is the, and after that, it also follows the, relatively quiet period of the firing. Okay, so that's the sort of the, this particular neurons 133's response property with respect to the monkey's report during binocular rivalry. And remember that this monkey is extremely precise in the eye movement, and sometimes they break the eye movements here. But most of the time, it's very, very precise. Okay. So when they averaged all the responses of neurons with respect to the time when they when monkeys started to change the lever pressing at 0, then many of the neurons, roughly 20% of the recorded neurons, started to increase the firing and then sustain firing when the transition was from non-preferred into preferred. Okay, and then the opposite is true for the preferred to non-preferred. So this is a kind of the pattern of the result that we can make sense of. On the other hand, roughly same proportion of neurons, like 20% of the neurons, showed this. So here, non-preferred stimulus to preferred transition happened, and here, preferred to non-preferred happened. But then the firing rate actually showed almost opposite pattern to this, nice neuron in a sense. We still don't know why, but this is a case for the B4 neurons as well as the MT neurons as well. But this is empirically found in the monkey experiment. On the other hand, if we go into the IT neurons, then IT neurons tend to fire really together with what monkey reports. So here, one particular IPD neuron prefers monkey face, okay? And then it starts with this sunburst pattern. So this is a percept and also eye input to left eye and also right eye. And the same goes for the other, format. The vertical tick is firing the neurons, and then this line is sort of a firing rate, smooth version of this line. And then at the bottom, there is a report of the monkeys, lever press. So it goes from patterns to some kind of, mixed percept where monkey was trained to release both buttons. And then, again, during this binocular rivalry period, monkey held initially this sunburst pattern and then switched into face perception, and then there is this increase of the firing just before pressing the button, and then after when it switched to pattern, it diminished the firing and so on. And then here from this point on, both eyes were presented with a face, so there is a huge increase of the firing for the face. Okay, so that's a more understandable kind of pattern of firing. In a sense, IT neurons seems to be very strong candidate of the neural correlates of consciousness. And this, the Logothetis and the colleagues did this type of the experiment a lot across V1, V2, and then V4, and then MT using the moving stimulus like this way, and then IT. Using faces versus sunburst pattern or something like each of the neurons prefer the stimulus in this case. And then there are many things to be told, but first, within V1, V2, only 20% or 10% of the neurons tend to show the firing pattern that is in a way correlated with preferred or non-preferred stimulus. In many of the neurons, it just stays the same, almost like reflecting the input from one eye constantly. The monkey's report doesn't actually make a difference in terms of the neural firing rate, okay? But that's the first observation. And then in the case of V4 and MT, as I said, roughly 20% of the neurons goes with what the neuron's preference, and the other 20% of the neurons go in the opposite direction. So when neuron prefers one stimulus, and then monkey report seeing that stimulus, it goes suppressed. And so this is an enigmatic kind of responses. And then finally, these IT neurons, they are almost 90% of the neurons going together with the monkey's percept. So that's the conclusion of this 98 paper. And the further research was done throughout the brain, going from So this is a summary in 2013, going from eyes to first thalamus, and then this is a LGN. LGN using the two types of the different stimulus, binocular rivalry and the general fresh suppression. Almost like less than 10% of neurons were related to the percept. Okay, and then if you remember, LGN to the B1 is not the only pathway, but the superior-colliculus to pulvinar is another pathway, and then there monkeys report tends to be more correlated with percept. And then B1, B2 is roughly 10 to 20%, and then B4 starts to become more highly correlated, 20 to 50%. Here, this figure doesn't discriminate between anti-correlation or correlation, and MT or MST, again, around 20 to 50%, and the parietal neurons are also similar, 50%. But these neurons in IT or STS, they tend to correlate really well, 100%, or 50 to 100%. And then FEF and lateral prefrontal cortex, they also tend to correlate with the percept at very, very high accuracy. That's a sort of emerging picture of the NCC research using binocular rivalry and the related phenomena. And here, this is motion-induced blindness, but the similar kind of story, basically. So, in summary of this section, what I said was that the monkey can be trained to report their conscious perception during binocular rivalry, and their psychophysical evidence is highly compelling, and there's a little to doubt that monkeys are experiencing binocular rivalry. And then extensive recording of the neural spiking revealed that NCC is unlikely to be found in the early visual areas such as B1 and B2, and that corresponds to B1 hypothesis of the Crick and Koch, and what I reviewed this week's at earliest video. And the higher visual areas such as IT and prefrontal cortex tend to have a really high ratio of the neural correlates of consciousness or correlated neural firing. And then at the middle level visual areas like B4 and MT, there is a mixture of the neurons. So what do they tell us about the NCC? So one thing we can say is that if IT neurons are the NCC, it's a bit weird if you think about it. And one of the reasons is that the IT neurons have a bigger receptive field, and their responses are invariant, meaning that as long as it's a preferred phase, it doesn't matter whether it's over there or there or there or there, or rotation invariance or something like that. Like Jennifer Aniston neuron, as long as monkey or human perceives that as corresponding to one concept, then it seems to fire. Okay, so it doesn't really corresponding to our conscious phenomenology. And also, meaning of the anti-correlated firing is also weird, at least, right? So all of these and other types of the finding, and also from a theoretical point of view, points to consideration of the NCC, neural correlates of consciousness, not as a single neuron, but as a population of the neurons. And when we start to think about the population of neurons, then the story becomes very complicated. And we are going to see the consequence of this population neuron coding type argument from the next week, starting from the binocular rivalry experiment in fMRI. And that's for now, for this week. See you next week.